Good evening, sir. You can start now, sir.
Yes, ma'am. Thank you. Thank you so much. Good evening, my dear students. Today, what we are going to discuss about any ideas? Last class, what we have discussed? Anybody else? So, what is by test cross? What is by back cross? That things already we discussed. What are the monohybrid test cross and dihybrid test cross? What will be the ratio after hybridization? Already that things already we discussed, isn't it? All are we will get 1:1 ratio only, isn't it? So parental character as well as which one the crossing characters also, the offspring also will get it a equal proportion, 50%, 1:1 ratio. That things already we discussed, isn't it? Okay. Then what is by back cross? What is by test cross? Back cross is nothing but what is that? F1 hybridization with any one of the parent, that is called as back cross, isn't it? Test cross means only recessive parent. So both we will get which one the 1:1 ratio only. Already about that things. Okay. So dihybrid cross is also same. So these are the F1 hybrid, F1 offspring, you will get the gametes. It's crossed with the parental, I mean F1 to F1, you will get it the 1:1 ratio only. Don't forget, my dear students, when we cross with the hybridization with the any one of the parent, that means test cross or back cross, you will get the ratio 1:1 only. Okay, there is no confusion. Okay, these things. And then, so what is by a trihybrid cross? Already we discussed three pair of characters, contrasting characters also cross together. As a result, what will be the product? Already we saw it in checkerboard. 27, 9 plus 9 plus 9, 3 plus 3 plus 3 plus 1. It's total 64, I think. Okay. So this is very, very important. So already we saw it, that things is also. Okay. And then, and chromosomal theory, who stated the statement? Walter and Sutton and Theodore Boveri, Boveri, whatever it may be. Pronunciation will be differ, but scientist name is Walter Sutton and Theodore Boveri. He stated the statement is called as chromosomal theory of inheritance. Okay. Okay. So these are the scientists you are seeing. Okay. So next, very, very important things, the TAM factors, note it down. This is very, very important. Replaced by Johnson in 1909. It's named as a gene. Okay, that things also already we discussed. And then we will pass on to the allelic interaction and Mendelian deviations. The allelic interaction they have given us an examples. Incomplete dominant, already we saw it a little bit in this portion. So incomplete, what is by dominant? Already know about this. Suppose any one of the plant we hybridize it itself, what happened? The dominant character will be expressed in the F1 generation, isn't it? But incomplete dominant means, what is that meaning? See that sometimes two allele of the gene do not show a dominant recessive relationship, but when present simultaneously or come together, they show an intermediate condition called the incomplete dominance. Already we saw it, that things also, isn't it? Which one is an examples? Already in our Mirabilis jalapa, I raised at 4 o'clock plant. This was a, what is that? Crossed by the name of the scientific known as, tested by Carl Correns, 1903. Okay. He got the sample. This is the plant already. I told you the Mirabilis jalapa, 4 o'clock plant. That things also already we saw it. Now, another example is also given, Antirrhinum. No, don't forget it. Antirrhinum majaz, otherwise called a snapdragon. This is also another type of plant. Okay. So it has the ability to incomplete dominant. It can cross there. You will get the option or answer. What is that? Incomplete dominant oriented. Okay. And then Andalusian fowl. This is also another one. This is very, very important. That things we are going to see. See here. So this is the snapdragon, otherwise called as dog flower. The dog flower, otherwise, what is that? The botanical, sometimes they were asked the questions. Snapdragon. Snapdragon is otherwise called as dog flower. And otherwise called as Antirrhinum mojas. And 4 o'clock plant also. Who stated the statement? That things also, you note it down very clearly. Okay. So what they did, see that in Mirabilis jalaba, the 4 o'clock plant, marvel of Peru. So this is a place. And the Antirrhinum mojas, snapdragon and dog flower. There are 2 types of flower color in pure state, red and white. The pure state means what is the dominant character as well as the another one. So here we will not tell it which both are dominant, isn't it? So incomplete dominant only. So both the character will be expressed in the F1 generation, isn't it? That's why the red is also a dominant character, capital or whatever it may be. A white W, small w or small w, like that. Okay. Okay, what happened here? A cross between a plant pure for red flowers and a plant pure for white flowers. Okay, yields the hybrid plant with pink flowers. This is very, very much in the F1 generation. What will be the dominant character here? Red is the dominant. So the F1 generation, you will get red only. But instead of red, what you got it? Pink flower. It is a mingled one. Okay. See here, neither red nor white is completely dominant. So that both colors appears in the hybrid as a blend, which is pink. I already told you. When you are crossed with the red and white, dominant character will be expressed in the F1 generation. Instead of red, what happened? The pink color expressed in F1 generation. Again, but just like a monohybrid, F1 cross with the F1. Okay, that pink flower again cross with the pink. What will be the F2 generation? See that? So when the 2 of hybrids plants with the pink with the red, pink and white flowers in the usual Mendelian ratio of 1:2:1 ratio, you will got it after the F2 generation. So here, see a 25%. This is considered as a 1 is denoted as 25%. 2 is 50. This one is 25. So total 100. Okay, you can calculate like that. Okay. 1% pure red. So 25% similar to that of the parent. 50 is a pink color, isn't it? 50 is the pink hybridization, incomplete dominance. The last one is also a 25%. It is what is that? White in color. See the cross, you can easily identify. See, so this is a red color. They can be denoted as capital C R and capital C R. This is the dominant character. Okay. But white color CW, CW. W is denoted as white. Here the R is denoted as red. Excuse me. What happened here? So corresponding gamete will be produced. See here, the capital C and the R is also expressed here. Gametes also produced. Similarly, the white flowers also produce the CW. The combination of the C, these two, what happened? The pink color will be expressed. CR, CW look like a pink color. It is not similar to that of the parent red, or it is not similar to the white. So then again, what happened to the similar things? Again, cross with the same self-pollinations. Okay, already you know, but F1 into F1. So you will get the 4. Okay, the 4 similar to that of the mono hybrid cross. 1 is a red and 2 is a pink color and 1 is a white pure in color. Okay, so here this is the 1% is 25% of the red, 50% of the pink, and 25% of the white color. That is the phenotypic ratio, phenotypic physical. Same thing he has given here. Another slide also. See here, This is the red, this is the white. What will be the colors here? F1 generation, all are pink color only. After that, cross with these two, you will get it a parent as well as a hybrid component as well as both the character of the parent will be expressed in respectively. Okay, these are the incomplete dominant. See the another one also. So red is the and white is the. These two are crossed. You will get the intermediate component of the a product of the pink colors. Okay, this is the checkerboard. When you are putting the checkerboard, it will be easy to find out the colors. We cannot be able to do any mistakes. Okay, so this is very, very important. You have to practice it once again. Okay, this is very, very important. See that same things also given. Red and white, you will get F1 generation is a pink. Heterozygote, this is homozygous. This is also homozygous. Here, there's both the character expressed here, it is considered as a heterozygotes. Selfing, you will get these are the type. This is the checkerboard. So phenotypic ratio, 1 red, 2 pink, and 1 white. Genotype ratio, capital R, see here, R1, 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 R2, 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 like that. They mention here the R1 is a dominant, the red color. R2, R2 is a white color. Intermediate R1, R2, pink color, like that. We can assume ourselves. Okay. Okay. Incomplete dominance is also found in the Andalusian fowls. The fowl is found in the 3 colors. In this category, these flowers contain how many colors? There are 3 colors have been identified by the scientist. Okay, one is called as black in color, another white, and blue is also present in it. These are the 3 colors identified from where? Andalusian fowl. The pure form are black, so they may be mentioned in the capital B. And the white, it is denoted as small b. So when you are going to cross between these two, what happened? The product will be whether the dominant is a black, means the F1 generation is also a black in color. But instead of black, what they obtain, the blue color obtained, because the combination of these two, black and white, you will get blue. If these two forms are crossed, F1 individual appear blue colored. The blue hybrids are crossing with each other. That is F1 into F1. So this is the F1 into F1. BP. See here, blue color and blue color. Likewise, a 4 o'clock plant. There, what is the pink cross with the pink? Likewise, here the blue cross with the blue. Because F1 generation we will obtain which one? Which color? Blue color only. The blue is blue, gives rise to the 1 black, 2, what is that? Blue, and 1 white is also there. That is called as 1:2:1 ratio. Similar to that of the, which one? The incomplete dominant, like as a mirabilis jalaba. Okay. So note it down. This is my dear students. Any doubt in this area, my dears?
No, sir.
Okay, go ahead. Just to follow it, it is better for you. See here. So this is the black. They denoted as capital B, and also white, small b, small. It is a parent. Okay. So corresponding gamete will be produced. Here the 1 gamete. That is a small b we will obtain here, and the black there is a capital letter is B. Okay. As a result, the F1 generation you will get it. Which one? This is called as G1 phase. That is considered as a gamete phase. Parental phase, gamete phase, the F1 phase. This is considered as a filial generation. Okay. So you will obtain both the combination of these two, the black and white. You will get blue color, blue BP. So again, it's crossed with the blue. What happened? F1 into F1. You will obtain the F2 generation. See, these are the combinations. Okay, 1 black, 2 blue, and 1 white. Already we saw it. This is also obtained from the Andalusian fowl genotype. Okay, so 1:2:1. 1 black, 2 blue, 1 white. Okay, this is also a incomplete dominant examples. Don't forget, my dear students. Now you studied 3 types, okay, 4 o'clock plants as well as the under cell fouls also. Okay. And the next we'll pass on to the a very important, it's called as codominant, also called as blending alleles. Sometimes they may ask the question directly. Blending alleles, also called as codominant. What is meant by codominant, my dear students? What is meant by codominance? Ajay?
Sir, both the alleles are dominant, sir.
Okay.
They both show the same, sir. Both the alleles, they show their respective traits, sir.
Very good. Super. So both the alleles, they express themselves, isn't it, in the next generation? Okay. Anyway, the codominant is a type of dominance where the offspring shows similarity to both the parents. Note it down, my dear students. Dhanalakshmi also answer it, please. Dhanusri and the others also. Both the parental characters also express in the offspring F1 generation. And it is due to the blending of alleles, blending. Okay, that's why it is called as blending alleles. Codominant is also called as blending alleles because it's expressed both the parental characters. This is very, very important. Okay, don't forget, my dear. Besides incomplete dominance, alleles could also show codominance here in a heterozygotes. What is by heterozygote? Both the character, isn't it? Both alleles express themselves. Therefore, the F1 generation resemble both parent. F1 generation always look like a parent, but it's not single. Both the parent character will be expressed in the F1 generation. Okay, see what is the example? I already saw it. So this is the examples, codominant and multiple alleles. They have chosen the 2 types of what is that? Cows or cows. Okay, take an examples. One is denoted as white, RW. Okay, this is the white color. Another one is called as red. So R, smaller, or smaller, like that. Both are crashed. What happened, you know? So it's expressed in the both the characters. See here, the white color is also present here, and also red color patches also present in here. That is called as Royan. Capital R, small r, and capital R, a small w. Both are expressed in the W. Okay, see here. So both the characters similar to that of the parental. Suppose when it's crashed with the rho n into rho n, F1 into F1, what will be the proceed? What will be the end product? You will get 1 of the parent, isn't it? 2 of the combination, F2 generation. And 1 again is the red also. White, this one is also 2, this one is also 1. So 1:2:1 ratio, you will get it. When you are going to put the checkerboard, you can analyze the ratio. Okay, see that. And another important thing is that you note it down, very, very codominant is an example, ABO blood group in human beings. Will you please tell me what is meant by multiple alleles? Okay, multiple alleles. Sometimes they may ask the questions, multiple alleles, given examples, ABO blood group. Ajay, what's your blood group?
Sir, B positive, sir.
B positive, always, okay. Okay, anyhow. So one more is that, what about Dhanalakshmi? Hello. So what about Dhanusri? Okay, anyway, B positive always one thing, Ajay, I tell you. Okay, listen. Those who are all are scientists, take their blood group. All are B positive. Go and check it, you will get the ideas. Okay, Ajay. So you
Yes,
should,
sir.
yeah, good. So ABO blood group in human beings, listen here. The ABO blood group are controlled by the gene is I. Okay, the gene is I. Note it down, my dears. This is very, very important. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled.
Excuse me, sir.
Yes, Pa.
So what does I mean?
I denote as gene. It's nothing but the chromosome. It's a location. Okay.
Okay, sir.
Okay.
Okay, sir.
What about your blood group?
So O positive.
O positive. Okay, good. Okay, anyhow. Okay, listen here. The gene I is there. Okay, but different alleles also present in the single locus is called as multiple alleles. See here, what are the types of allele present in here? So many times they were asked the question. This is very, very important, my dear students. The plasma membrane of the red blood cell, RBC, has sugar polymer where the plasma membrane of the red blood cell. Okay, note it down. That protrude from its surface, and the kinds of sugar is controlled by the gene is called as I. The gene I has 3 alleles, already told you. Okay, I, A. The I is a gene. Okay, the allele is A. Here I is a gene, the B is a allele. This is a small I. This is also one of the alleles. See here. From this, the alleles IA and IB produce a slightly different from the sugar, while the allele small I does not produce any sugar. Note it down, my dear students. The blood group, when you are going to discuss about the blood group, it is also one of the codominants because the gene I contain 3 alleles. This is also called as what is a multiple alleles present in the same locus of a chromosomes. The IA, IB, they produce the which is a different form of sugar molecules. But I, small I, it will not produce any sugar molecules. Note it down, my dear student. This is also asked in the NEET questions. You got it, that points? The gene I is it is present in the plasma RBC. How many alleles are present in IA, IB, I also the small I. IA, IB produce a different form of the sugar molecules compared to the small I. They will not produce any sugar molecules. Are you clear? Okay, go to the next one. Okay, because humans are diploid organisms. Each person possesses any two of the 3 I genes alleles, isn't it? I genes alleles, because we are all getting the blood group. Suppose take an examples, Ajay is blood group B. Okay, what will be the phenotype? What will be the genotype? We must note it down, isn't it? We'll see one by one. See, the I is the normal one, but A and B and small I is the alleles, 3 alleles, isn't it? Here, see here, IA and IB are completely dominant. Listen here, A group as well as a B group blood. It's a dominant blood group compared to I, small I. Small I denotes what is that, O group. Okay. In other words, so once again repeat for you. The I gene contains 3 alleles. One is A, one is B, one is small I. When you are going to write, what you have to write, with addition of I also. IA. This is the gene, this is the allele. Here IB. This is the A blood group, this is the B blood group. Okay, IB. I is a allele, sorry, I is a gene, B is a allele. Here you have to write which one, small I only. So in other words, when IA and I are present only, IA is expressed. Okay, IA is expressed. What is the meaning? Suppose IA is here. Which one is expressed? The I is also present, but IA only expressed. But IB is also similar to that of the IA. When I is present in the IB only expressed. Okay, this is the category. This is the dominant characteristics. But when IA and IB are present together, they both express their own type of sugar because of codominant. What you are knowing here is we know very well about this point. IA is nothing but the A blood group. IB is a B blood group. IA, IB, suppose if it is present in the single chromosome, what happened? The blood group will be AB. Okay. Do you know that here this is considered as a codon? Those who are having the blood group AB, they are all comes under the heading is known as codominant group. IA and B are what is that? Dominant group. I is a recessive group, otherwise called as O group. And IA, IB both are present in the single chromosome. They are called as codominant group. Okay, the blood group is AB. Okay, don't forget, this is very, very important. Hence, the red blood cells have both an A and B types of sugars in this category. AB blood group, they contain which one you know? AB type of sugars. A blood group only presence of A sugars. B group only B sugars. But I group, there is no sugar molecules. Since there are 3 different alleles, there are a total of 6 different genotype of humans, A, B, O blood types. We'll see these things, isn't it? Excuse me. See that? This is the genotype. This is the blood group. Already we discussed clearly once again. The I is gene, isn't it? I is a gene. This is called as alleles. When both the character is present, IA, the blood groups must be A. Because both the characters, both alleles are same. So the blood group is also same. Here, see here, heterocytes. IA and IB, both the sugar molecule present. What about here? Only A sugar molecules only present, A type of sugar. So A. Here, both the characters present, A and B. So the blood group is AB. It's also codominant. But what about here? IA and small I. Small I is a recessive. Which one is expressed? Dominant character only. So A is there. Here, IB, homozygous, B sugar molecules only present. So this is called as B blood group. And then IB and I, small I is present. Small I is not expressed. So the B is expressed here. The B is. So Ajay, what about your blood group genotype means? Which one is that? These two only, isn't it? My dear.
Yes, sir.
The IB and IB. And
Yes,
IB
sir.
and small I, isn't it? These two only.
Yes, sir. IB and IBI.
Yes, yeah. Suppose if you know your father blood group, you can easily identify which genotype you are. Okay. Okay. Dhanalakshmi. Yeah. Which type you know? Small I. Because we are all sorry. I also the yeah. O negative. O positive.
Okay, sir.
Okay. Small I denoted as O type. So this is the recessive. Okay. Which one is a universal donor, my dear? Which one is a universal donor? I ask you one question. Which one of the following is a universal donor?
Sir, O positive.
Who said?
Sir, Dhanalakshmi.
Okay, anyhow. O positive is correct or not?
So universal donor is O positive only, sir.
Sir, I will tell you once again. I ask you one question. Just listen carefully. Okay. Which one of the following is a universal donor? A, AB, O positive, and O negative. Which one is correct, my dear? Dhanalakshmi. Ajay, do you have any idea about this? You don't know, isn't it? Ajay?
Yes, sir.
O negative. Because when you are going to see the blood type, you know, see the next slide, you can understand. See here. So blood group, isn't it? Phenotype. Blood group A, blood group B, AB, and O. So A means you know about IA and AA, genotype, isn't it? Otherwise IA and small IA, isn't it? So these are the two type is possible genotype. So what will be the antigen? You know, blood group, which one is that? There is a corresponding antigen will be present. The antibody will be differ already you know about. So anti is the blood group. This is called as genotype. This one is called as antigen. So antibody is always opposite manner. B is present. So it is a nature is dominant. Already we discussed. Okay. Okay. So they will give. What is the last column? The transfusion accept from the A blood group to A blood group or O blood group. isn't it? They will not get from the B. Suppose we are getting the blood group B as well as the AB. What happened? Agglutination is possible lead to death. Okay, because antigen A, okay, react with the antibody A present in the B blood group. See the B blood group, the antibody, antigen is B, antibody is A. These two are react together, and likewise these two are react together. As a result, the formation of agglutination lead to the death. So blood transfusion, when you are taking blood transfusion for any other process, take an examples of Ajay, you must take a blood group from which one, B or O only you have to receive blood group. Okay. And then what about the O? O only receive from the
So,
O
O,
group.
sir.
Yeah, very good. O only. Suppose if it is negative, very rare, isn't it? O negative means very rare. Okay. In millions people, there is a 1 or 2 only, 10 or like that. So that is a very complicated one also. Sometimes, about there are so many ads also coming. You saw it in the net or something else, your cell phone also. The B positive, B negative blood group is very, very essential for the so and so like that. They comes. Okay. So that's it. Leave it. See here, once again repeat for you. The blood genotype A and B, AB and O. The genotype already we discussed 6 type of, how many types of blood group is there? 6 type of genotype we can obtain. Once again repeat for you. How many blood group is there? A and B and also O only is there. But we will obtain how many genotype means 6 genotype. 1, 2, 3, 4, 5, 6 genotype. This is also so many times they were asked the questions. Okay. Antigen already know. Antigen A, sorry, blood group A, antigen A. Blood group B, antigen B. But antibody is different. A antigen, A, B antibody. B blood group, B antigen, A antibody. So both are dominant. So suppose if it is A, B, so both A and B is also antigen. Neither A or B is present. This is called as codominant. Now we are studying, isn't it? But the agglutination is possible, none. So they can receive any type of blood. Okay, A, B, AB or O. That's why this group AB is denoted as universal acceptor. Universal donor is a O negative. Suppose the question paper, you must note it down. Suppose O negative has given, don't forget O negative is the answer. Okay, don't tick the O positive. So this is very, very careful. Just carefully read, then only answer it. Okay, this is the things. Then ABO blood group, these are the things already we discussed when you are going to discuss the agglutination process, immunology. Okay, that time we will study. This is the inheritance character. Suppose the parent A is the parent B, father or mother, whatever it may be, you can change it, whatever it may be. Child denote is AA, you will get the A. A and O, AO, you will get A. So A and B, AB, it is nothing but the codominance. B and A, AB though. So AB is there. It's also codominant. Suppose B and B, BB. So you will get B. B and O, BO. So this is a B. Because the O is a recessive. So it note it down, very, very important one. A is a dominant character, O is a negative. Okay, recessive characters. So OO is a already in our ability. When in the recessive character, both alleles are expressed together, then only it will be comes out. That is the reason. So the parent is also the mother as well as the father is also OO. The child genotype is OO. The blood group is also O type. Okay. So this is very, very important. Sometimes they may be likewise. This one. Okay. So this is very, very important. Don't forget, my dear students. Any doubt in this area, my dear? Here, so many times is the blood group A, B, AB, O. Genotype 6. How it will be expressed? Already know. Antigens, antibody. Which one is the dominant character? Which one is the codominant character? Which one is the recessive character? Okay. So transfusion acceptors A or O, B or O, A, B, AB, and O, and also O. So this is the parental genotypes. Okay. Child blood group is also mentioned. Okay. Next one is very, very important thing. Coat colors in short horned cattle. There are two types of pure short horned cattle, red and white. See here, red and white. On cross breeding, the individual of the F1 generation are found to have a roan color. Already discussed. The effect is produced due to the, what is that? Juxtaposition. This is also given as a different technical term were used, scientific name of small patches of red and the white color. Sometimes you can see the white color cow is there, but red color patches is there. Sometimes the red color cow is there, the white color patches is there. Such type of what is that? Hybridization is known as mosaic inheritance. There is a dot, patches is denoted as the mosaic inheritance. Note it down, my dear student. It is also known as a mosaic inheritance. The mosaic inheritance also called as which one, you know? Co-dominant. So sometimes they may ask the question likewise. Okay, see that? So coat colors. Okay, this is we will discuss later. Okay, here the wild type brown fur, black tipped white fur. Each one it has a specific alleles. The CH denoted as chinchilla, black tipped white fur. Okay, fur means you know what, it's nothing but the hair covered by the skin. CH, this is Himalayan white fur, white with the black paws, nose as tail. These are the another types. Himalayan types, chinchilla type, wild type, and albino type. Albino is nothing but albino. Guinea pig is also one of the albino. Rat is also present. These are all mostly used for the laboratory purpose. Okay. C, capital C, small c, albino white fur. The genotype they may ask the questions. Himalayan white fur, which a genotype is present. Capital C, small h, capital C, small h. Here, chinchilla. Chinchilla means what is it? CH is a small one. CH. CH. These are the characters. Capital C is denoted as wild type of brown fur. These are the alleles. Okay, and genotype is also possible. So blood group already we discussed. Blood group possible genotype formula N into X. N plus 1 divided by 2. How many blood group is there? Already in our A, B, and O. So 3 types. How many genotype is possible? In our 6 type of genotype is possible. Already been discussed. Coat color of rabbit is, see here, N into X, N plus 1 by 2. How many coat color is there? 4 types is there. See here. 1, 2, 3, 4 types. So 4 means you have to calculate in the apply the formula. What will happen? 4 into 4 plus 1 divided by 2. So 5. Here is also 4. So strike out this one 4 up to this one 2. So you will 2 into 5 you will get 10 genotype. See here. So these are the types. 4, 3, and 2, 1. The brown, how many genotype is possible? Capital C, capital C, capital C, small c, CH, capital C, small c, H, capital C, small c. The capital C only expressed it. That's why its color will be brown in color. Okay. Light brown. See here. 3 only possible. CCH is possible one. CCH, CH is possible. CCH, C only possible. This is called as light brown. Himalayan 1 is 2 is possible. CH, C. Okay. So this is called as what is that? Himalayan is possible. Last one is called as albino. This is small c, small c. These are the 10 possibilities is also where. How will you get it? When you are going to put the Punnett square, you will get this type. See here. And sickle cell anemia. And next one is an important thing. Sickle cell anemia is a disorder in which afflicted individual contain red blood cell that become elongated and curved under low oxygen tension. Normally, you know about the oxygen is essential for the metabolism in our body, anywhere, any part of the body. It is one of the connective tissues, blood. RBC carry how many? Each hemoglobin carry how many oxygen molecules? Will you please? How many oxygen molecules carry each and every hemoglobin? Any idea, my dear?
Sir, 2, sir. I'm not sure, sir.
No.
Sir,
What about
4.
that?
4.
Yeah. Correct. Exactly. 4 molecules only. Okay, 4 oxygen molecules carry out by each and every hemoglobin. Don't forget, my dears. Okay. So elongated and curved one. So the oxygen is essential for the maintenance of the structure of the RBC. So suppose if it is any physiological or chemical denaturation happen, what happened? The RBCs look like a sickled shape due to the low oxygen tension. Okay, such sickling of erythrocyte does not occur in the normal individual, and they retain a biconcave disc shape. Okay, individual with this disease suffer attacks due to the aggregation of the red blood cell, the capillary system of venous side, and several tissues suffer severe damage. How it will happen? Suppose when you are taking any one of the tablets or drugs or whatever it may be, what happened in it? It will be damage of the RBC. Okay, unwanted materials. Take an example. Say any one of the microorganism, alien species, or foreign particle like as a virus, a bacteria, whatever it may be, any other foreign particles due to the harmful effects, it will be denature the oxygen. Denature, not only the denature, damage of also occurs. As a result, what happened in the RBC normal shape will be changed into sickle cell. That condition called as sickle cell anemia. Okay, the oxygen also known as the sickle cell crisis, because suppose when it is changed into the normal RBC into the sickle cell, they will not carry the enough oxygen molecules from the one place to another place. As a result, the wheezing problem and the body temperature, it will be rise. Morphological symptoms also occurs within the body. Okay, see that. So this is called as, what is that? Sickle cell. See that this is the normal red blood cells and contain hemoglobin molecule. It carry the oxygen molecule from the one place to another place. Suppose if it is the normal one, change into the sickle cell type. Sickle cell, see that sickle. Okay, they used for the harvesting of rice or wheat, like as a small sword-like structures. The abnormal hemoglobin, inside there is a abnormal hemoglobin. This condition is called as, what is that? Anemia, sickle cell anemia. Okay, due to the chromosomal aberration. We will study that things also. Okay, all these erythrocytes are destroyed more rapidly than the normal red blood cells, leading to anemia. This is controlled by a single pair of alleles. Note it down, my dear students. Hemoglobin A and hemoglobin S. This is also so many times they were asked the question. The sickle cell anemia disease is controlled by a alleles is called as which one of the following? HPA, HBS, AS, HB, hemoglobin A, hemoglobin S. Pedigree analysis revealed 3 genotype of 2 phenotypic with normal and affected individual. When you are going to analyze this, suppose any one of the persons suffered by the, what is that? The sickle cell anemia, how it will be? We can put the pedigree analysis. We go through the pedigree analysis. We will find out the phenotype ratio, normal versus with affected individual, resulting from the homozygote genotype HPA. See that HPA and HPA and HPS, and the last one is HPS respectively. Heterotype of genotype already in our HPA, HPS exhibiting sickle cell. So yes. So these are the, what is the genotypes possible is the pedigree analysis. These are the things is there. So don't forget, my dears. And then multiple alleles already we discussed. More than 2 alternative form alleles is called as, what is that? Alternative form of gene is called as alleles. Suppose if it is present in the a single locus, more than 2 alternative form of alleles. Alternative form is nothing but more than 2 alleles of a gene in a population occupying the same locus on a chromosome or its homologue are known as multiple alleles. It's nothing but single loci contain more than one alleles. That condition is called as multiple alleles. Already discussed. The all multiple alleles occupy the corresponding loci in the homologous chromosome. Okay. See, this is the locus is nothing but the place, occupying place. Okay. In any diploid individual of only two alleles can be found. So multiple alleles can be detected only in a populations. Multiple alleles express different alternative of single triads. This is also a straight questions. Multiple alleles express different alternative of a single triads. Different alleles may show codominance, dominance, recessive behavior, or incomplete dominance among themselves. The different alleles present in it, that alleles, each one it has a specific properties. Sometimes they show codominant characters, dominant characters, recessive character. Sometimes it may be a incomplete dominance. Okay, they can express. We cannot be able to say which this one is a dominant, this one is no, whatever it may be. Each one it has a specific property. See here. So these are the examples has given for your multiple alleles. Here ABO blood group already we discussed. Gene has three alleles, IA, IB, and I. So this is the first one. Histocompatibility, gene complex, HLA complex. When you are going to study antigen antibody activity in human, okay, so that time we can study HLA. You note it down this time. So many times they were asked the question. This is also here. HLA expansion, histocompatibility. Okay, so and then self-incompatibility in plant, self-incompatibility in plant, and then eye colors in Drosophila. Okay, the eye color in Drosophila gene has 15 alleles. How many alleles present in it? 15 alleles. Sometimes they may ask the question, Drosophila, the eye color, it contains how many alleles? 15 alleles. The coat color of the rabbit, already we studied that things also. How many alleles is there? 4 alleles. Okay, don't forget my dear here. Multiple alleles, these are the examples. ABO blood group, histocompatibility, self-incompatibility, eye color in Drosophila, coat color in rabbits. So corresponding alleles, how many alleles also present? Suppose if you want to know, here are the 3 alleles, isn't it? So these two, if you want to know, just go through the Google. Okay, you will get the idea. If you want more information. Formula of genotype in multiple alleles already you know, but n by 2 into n minus 1, where n is the number of alleles. You will get the answer. Okay, 3 means you will get how much? 4 means how much? 15 means how much? That things we may find out. Already we saw it, these things, isn't it? Multiple alleles I have given as a examples. Okay, I think this one is I finished it. This is part 2. Okay, so today what we have discussed already in our report. What are the things already we discussed? Will you please? Incomplete dominance. Okay, incomplete dominant. They have told which one you know. So one is called as 4 o'clock plant, Mirabilis jalaba. Another one is called as Antirrhinum majus. So what are the combinations? Red and white is the, and how it will be the F1 generation, pink color. Okay, that things. The Mendelian triads is also what happened here. 1:2:1 ratio will be obtained due to the incomplete dominance. So checkerboard already in our report. Likewise, which one is also the Antelsian false. Here 3 color has mentioned. One is black, another one is white. You will get it. Blue color in the F1 generations. So final result similar to that of the incomplete dominant. 1:2:1 ratio. So another one already we saw it. This Antelsian false genotype. Co-dominant, otherwise called as blending alleles. Both the parental characters also expressed in the F1 generation. F2 generation, you know, 1:2:1 ratio. One is a red, 2 is a drone, and another one is a white color. here you note it down, my dear student. The roan color phenotype, just a capital R, small r, capital R, small W. This one is so many times they were asked the question. So don't forget, my dear. Capital R, small r, capital R, capital W. These things. So the roan, okay, the F1 offspring, the genotype, characteristics. Okay, sometimes they may ask the question, which one of the following? Sometimes this one they may give, this one they may give. So this one is the, so this one is the combination of both. So don't forget, it is easy, I think. Okay, so ABO blood group already we discussed. 3 alleles is the A and B, and also I is also there. So AB is a go dominant. Sometimes they may ask the question, which one of the following blood group is a co-dominant? Which is the one, co-dominant blood group? Ajay?
Sir, IA and IB, sir.
No, I have to tell the fully. IA and IB, totally called AB blood group. Okay.
Yes, sir. Sir,
No
IA
need.
and IB, sir.
Yeah. Alleles. Gene with alleles you are telling. So yes, correct only. But AB blood group. Okay. Anyhow. So what about the dominant one?
Sir, both are dominant, sir. IA is also dominant, IB is also dominant, sir. But
Yes.
I small I is, sir.
Okay, good, super. IA, IB is separately dominant, isn't IA and IB. And also IA and IB together, it is also a co-dominant. Suppose small I is there, what is that? Negative. Otherwise called as what is a recessive. Okay, don't forget. Okay, good, super. Well understood. Okay, what are the next one? So these are the characters. How many genotype possible? 6. Don't forget, my dear. 6 possibilities there. They may ask the blood group. They may give the question paper. They may ask the alleles, genotypic aspects. A means what, B means. Tomorrow you must go through it, this one. Okay, you practice it, my dear students. This is very, very, very important one. Don't forget, my dear. Already we discussed it. Which one is the antigen and the antibody is also dominant, co-dominant, recessive. Already we discussed these things. Okay, transmission. Which one is the universal donor? Which one is the universal acceptor? And that things also. Okay. So sometimes they may ask the question, the parent as well as the genotype of child. Ask the question. So don't forget this slide, my dear. Okay. So go to the next one. And code colors. Already we discussed it. What are the 4 types of code color? Is the allele, genotype, how it will be expressed. Okay, that things also already we discussed these things. How many genotypes may possible? Suppose the color of the rabbit is N is 4, you will get 10. N is 3, you will get 6. Suppose if it is 15, how many? 15 plus 1, 16. So you have to calculate 90 genotype will be obtained. So you have to practice the more than similar to that of the question, just to go through the which one previous year question paper. Okay, these type of question only the max oriented. Okay, these type of question will be, they may ask the questions. Depends upon the what is the genetical aspect, gene aspects, genotypic aspects, allele aspects, and also the possibilities. Okay, see this one. This one also sometimes they were asked the question. Albino, small c, and then this one. Okay, light brown. This colors is also, how many possible is there? Brown 4, light brown 3. So you have to calculate here 1, 2, 3, 4. In the ascending order you calculate A 1, H 2, L 3, B 4. 1, 2, 3, 4. Like that you calculate it, it will be better. Okay, understand that one also. Okay, and sickle cell anemia already available. This is very, very important things. Okay, why it happens, how it happened. This is also one of the recessive characteristics. So it will not carry the oxygen molecule due to the abnormalities of the hemoglobin molecules. Changes occurs within the blood. Okay, so as a result, it not carry the oxygen molecules. Okay, this is also lead to the anemic conditions. What are the pair of alleles represented? Means HPA, HPS. Okay, this is homozygous. Heterozygous means both are present. Okay, don't forget, my dear. The multiple allele already we discussed. Okay, Each one it has a specific. What are the examples? ABO blood group and histocompatibility, self-compatibility in plant, eye colors in Drosophila. Then gene has 15 alleles. Here the 4 alleles also present. The gold color already discussed. That things also. Okay, so that's all about today classes. Remaining things we will see the next class. If you have any doubt, please ask me, my dear. So here I couldn't understand. So if you have any doubt like that, you ask me now. Do you have any idea doubt, my dear?
No, sir.
Okay.
No, sir, no doubts.
Okay, Kanas. So the next part is already you know about the principles inheritance of the variation, part 3. Just I gave you an introduction. I will finish it here. Gene interactions. Gene interaction is nothing but what is in one gene is also joined with the another ones, otherwise called as crossing over takes place. As a result, you will get the what is that variations. Okay, so gene interaction is nothing but the variations. Okay, so we are all are the what is a hybrids from the mother or father chromosomes. As a result, we are having both the characters, father as well as the mother characters. So gene interaction, how it will be there, that things. And then synthesis, epistasis, suppression is also here. There are, and epistasis also further divided dominant and the recessive epistasis, hypoepistasis. Okay, so these are the two things. And then so epistasis, there is a example is as given the coat color in mice. So already we discussed, you know, in just like a rabbit, brown, black, pigment, no pigment. So these are the epistasis, how it will be the phenotypic ratio as well as the genotypic ratio, all those things. Okay, and then dominant epistasis, how many phenotypic ratio is obtained, 12:3:1. What are the examples? Fruit color in summer quas, like that comes. This is the fruit quas. Summertime, what happened, you know, the white color is there, and also green color is also there, and sometimes the yellow color. What will be the genotype? See here, WG, W small g, W small w, capital G, and then here small w, small g, small g. These are the yellow color. Yellow color is the recessive. The capital is the dominant character. Okay, that things we will see the next classes. Thank you so much for your participation. Bye. Take care. Good night.
Thank you, sir.
Thank you, Paul.
Thanks, sir.
Thank you, Kanna.
Thank you, sir.
Thank you, ma'am. Thank you so much.